Hello, I am making this recording to share with everyone in class the concepts we explored during study group this week in Bio 12. And so there are two key concepts in chapter 23 that we need to have a good, solid understanding of what they mean. One is microevolution and how we use the Hardy-Weinberg equilibrium concept to decide if microevolution is happening or if a population is not changing. So let's begin with looking at the question, what is microevolution? When you look at the prefix micro, micro means small. So this is going to be changes, evolution at a small scale, and it's going to apply specifically to populations. What about a population can change? Well, one of the things we can measure and see if change or evolution is happening is going to be the changes in allele frequencies in the gene pool of the population. So all populations have a gene pool. The gene pool is going to be the collection of all of the alleles, all of the members in one population in one specific place are going to possess. And so it's like an inventory. Think about all of the alleles the members of a population have with regards to body color, body shape, specific abilities to digest certain kinds of food, everything that is part of the genotype of all of the individuals put together makes a gene pool. And what I'm saying is that microevolution can be measured if you can detect changes in the frequencies of alleles. So think, for example, about one specific gene at one locus. So the gene locus is going to be the specific location of a gene in a chromosome. For a gene locus, you may have, for example, two alleles, a dominant and a recessive. And so what I'm saying is if you have a particular trait of interest, like last week, we were looking at the trait of beak size in finches. This week, you're looking at coat coloration in rock pocket mice. And so think about one of those traits that are important for survival, and you want to see how the frequencies of the alleles responsible for those traits may be changing or not, then you can do a study to see if microevolution is taking place. So what can happen? Alleles sometimes can decline in frequency. They can become less frequent, or sometimes they can become more frequent. If they become more frequent, more members of a population show that particular allele. If it becomes less frequent, it could get to the point that an allele disappears because all of the members with that less desirable allele are not going to be surviving, and when they die, the alleles also go away from the population. And so think about one specific gene locus, two alleles for that locus, and we're looking also at only individuals in one specific place that are members of the same species, and that is going to be a population. You cannot be looking at what many different populations are doing. You have to focus only on one population. So what we know is that natural selection is going to lead to differential reproduction. What is differential reproduction? Differential reproduction means that in a population, there are going to be those successful individuals which produce large numbers of offspring. Some members of the population may produce a few offspring, but there will be many members in a population that will never produce any offspring. You can then deduct that those individuals that are successful in producing larger numbers of offspring are going to be passing more of those alleles that make them successful to the following generations. And that's one way microevolution may be happening as those alleles increase in frequency. In chapter 23, natural selection and its consequence, differential reproduction, are described as the main force driving the process of microevolution. So I hope you get an idea. Microevolution is we're trying to see if there are small scale changes happening in allele frequencies, in the members of one population, within a population. Now, related to this idea of microevolution is also the concept of Hardy-Weinberg equilibrium. Hardy-Weinberg equilibrium will be the situation where in a population, the gene pool remains frequent. The alleles you have in a population not only are remaining generation after generation, but also the frequencies of those alleles is not changing. The alleles that are always more frequent remain frequent in larger proportions, and those alleles that are less frequent keep their low frequency. If there's no change in the gene pool, then you can say the population is going to be at Hardy-Weinberg equilibrium. Think about it as a question that has two possible answers. And the alternative is going to be giving you the answer to something that you are rejecting. For example, if you say that evolution is not happening, then you have to accept that the population may be at Hardy-Weinberg equilibrium. Or if the population is not at equilibrium, Hardy-Weinberg equilibrium, then the population is going to be evolving. So it's a question for which we have two answers. It's one or the other. There are going to be no in-betweens. So now you have to think about what would it take for a population to remain constant with regards to its gene pool to maintain the Hardy-Weinberg equilibrium. And so biologists have identified 5 conditions such a population at equilibrium must meet. One of those is that a population must be large, so many individuals that if you say, hey, 100 died because of some catastrophe, or some got sick and they died, those that died don't change the gene pool of the population. The same frequencies of alleles remain. And for that to happen, you have to have many thousands, maybe hundreds of thousands of individuals in a population. And as we go down this list, I want you to think, what population can meet all 5 of these criteria? Because even if you have a population large enough, you also have to have a population where no gene flow occurs. Gene flow is just a natural consequence in nature, where competition sometimes gets too intense in a population and individuals begin to move out in search for better places, with more resources, with less competition. And as individuals begin to move out from the population, as those individuals are flowing out, well, the alleles they possess are also moving away from a population. So you have to think about a population where no gene flow occurs. Nobody leaves the population. It's like living in prison. All of the members of the population are locked up. Nobody can leave. Nobody can escape. Now it's getting harder. The population must be large. There must be no gene flow. Nobody can move out. Also, mutations, if they happen, they will have to be at the level of non-coding DNA. Or maybe if a mutation happens, it just replaces one DNA letter for another one, but you still end up with the same sequence of amino acids in a polypeptide. As you learn in Bio 211, we call those silent mutations. So mutations will be producing no new characteristics. Beneficial or detrimental mutations have no effect on the genotype or the phenotype. What else? Oh, for a population to be at equilibrium, the mating is going to be unrestricted. Finding mates, reproducing. Any member of a population has an equal chance of finding a mate and successfully going through the process of reproduction. And if that's the case, then we have to add 1/5 condition, and that is going to be that all of the members of the population would have to produce equal numbers of offspring. If you're looking at a population of animals, let's say that every member of that population of animals is going to produce 5 offspring, and the next one produces 5 offspring, and every member is producing exactly 5 offspring, no more, no less. And now you can begin to think, well, what population can meet all of those 5 conditions? And even though I wrote here on my slide, they are near these conditions, all 5 of them together are nearly impossible to meet. I'm not going to dare to say they are just impossible to meet, all 5 of them. If one of the 5 is not met, the consequence is going to be change, microevolutionary change, and Hardy-Weinberg equilibrium will not be maintained, will not exist. And so why do we have this idea of Hardy-Weinberg equilibrium conditions if nothing can meet them in nature? Well, I'll tell you, the concept of Hardy-Weinberg equilibrium is used by evolutionary biologists as a null hypothesis to say, okay, so what would it take for the gene pool of a population not to change from one year to the next one, from one generation to the next, no change. In statistics, the hypothesis of no change, no difference are going to be always null hypotheses. And so that's what the Hardy-Weinberg equilibrium is going to do here. Well, let's see how we can apply it then to test the hypothesis that a population is at equilibrium and not changing. So there are 2 expressions of this Hardy-Weinberg equilibrium we can use. One is going to be the frequencies of alleles. So here in this expression, P plus Q equals 1. P is a symbol for one allele, could be the dominant or the recessive. We don't have to identify that. So the frequency of alleles P plus the frequency of alleles Q must equal 1. And frequencies are real easy to calculate. In statistics, we do it by dividing a number of observations over the total number of events. And I'm going to be using examples to help you understand a little bit better. Another expression of this Hardy-Weinberg equilibrium is going to be what we call the Hardy-Weinberg equation. This is it right here. P squared plus 2pq plus q squared equal 1. Remember, P and Q are symbols for alleles. Now, what about P squared? P squared represents one of the homozygous forms you can have when an organism has 2 copies of the same allele P, and Q squared is also going to be a homozygous when a member of the population carries 2 copies of allele Q. And we also have heterozygotes. PQ is going to be the symbol we use for individuals in a population who carry allele P, and they also have a copy of allele Q in the genotype for this gene at one specific locus. Okay, with that information, let's take a look at an example similar to what you saw in your reading for this week's discussion, rock pocket mice, where you have a lava flow. The majority of mice are going to have a dark brown fur because the dark fur is an advantage when these mice live in a place where dark rocks are going to be the basis for the background in the environment. And so let's say we go and we make an expedition and we go to New Mexico and we go to one of these volcanic lava flows and we capture 200 rock mice. Out of these 200 mice we captured, and again, you can see how this is done in the video for this week's discussion, out of the 298 are homozygous for brown fur. So now we're looking at the data we obtained from the fur samples or blood samples we took from these mice, and we've analyzed that in the lab, and we know that 98 out of the 200 are homozygous dominant for brown fur. 84 also have brown fur, but these are heterozygous. And 18 have very light, almost white fur, and those 18 are going to be homozygous recessive for the white fur allele. So how do we get the genotype frequencies? The frequency of those that are homozygous for the brown allele can be easily calculated by dividing 98 over 200. and you come up with this number over here. If you divide 84 over 200, gives you the frequency for the heterozygote allele. And if you take 18 divided by 200, gives you the frequency for the homozygous white fur allele. I'm sorry, genotype. So these numbers you see here, in the next line, I'm explaining how I came up with these calculations. Divide 98 by 200, you end up with 0.49, which is what we have here. Divide 84 by 200, you end up with a 0.42. And I'm sorry, this should have been 18. 18 divided by 200, I have to make a correction here, equals 0.09, and that's going to be the frequency of the genotype homozygous recessive for white fur. I just discovered there should have been 18 here, not just 18. I apologize for that, but you can make that correction too. So, okay, you have to add these numbers and you're going to end up with 1. What about the allele frequencies? The allele frequency, I'm telling you, for the brown allele is going to be 0.7, and the allele frequency for the light color allele is going to be 0.3. But how do you calculate those allele frequencies? Based on the explanation I provided in my video, what you have to do is look at how many individuals are homozygous for the P allele. In this case, P is representing brown, and we have 98 homozygous. Each of these 98 carries 2 brown alleles. So we're going to multiply 98 times 2, the 2 brown alleles in each of these 98 mice. But we also have more brown alleles in this population. The heterozygotes each carry 1 brown allele. So we need to add 84 brown alleles for the heterozygotes in the population. That's going to give you a total of 280. And now we're going to divide 280 over 400. Why 400? We're dividing over 400 because we have a total number of 400 alleles. Look, we have 200 mice. Each mouse has 2 alleles for fur color. So if I ask you how many total alleles are we dealing with in this study of fur color in mice? 200 mice, each with 2 alleles. 200 times 2 equals 400 alleles. And that's the number we use over here. And here's how you calculate the frequency for allele Q, the white allele. We have 18 homozygous recessive, those that have 2 white alleles. So 18 times 2 plus the 84 white alleles we have in the heterozygotes. That's going to give you a total of 120 divided by 120, 120 by 400, and you end up with 0.3. And that's the value for the white allele. 0.7 plus 0.3 equals 1. And so that's the data we have for our original study. This is our baseline data when we begin our study. And then let's say, this is interesting. Now we have to ask the question, is this population evolving? It's one specific place, one rocky outcrop in the desert. That's the area of our study. We go back the next year and we capture some mice. But the next time around, we only captured 120. It's like these mice are getting smart. They're not being fooled by the traps we set up the previous year. And so next time around, we only get 120. I don't know. That's the number we get. It doesn't matter. And in the 120 mice we captured, we did the analysis, 65 are homozygous dominant for brown coat, 50 are heterozygotes, and 5 are homozygous recessive for white coat. And so now this is where you can practice, and you can grab your smartphone, use the calculator function, follow the steps I showed you in the previous slide, and see if you can calculate the genotype frequencies and allele frequencies. And if you're following this presentation, I would recommend you do this. Pause the video at this point. Don't continue watching the video, and see if you can, answer the question. what are the genotype frequencies, and what are the allele frequencies. Pause it right now, and when you are done with your work, resume it. And after a short moment, you come back and you can check your answers. The genotype frequencies are going to be 0.54 for homozygous brown, 0.42 for the heterozygotes, these are also brown mice, and 0.04 for the homozygous recessive, the white mice. And these numbers would be calculated by simply dividing 65 divided by 120, you get this number. 50 divided by 120, you get the number for the heterozygotes. And you take 5 divided by 120, and you end up with the number for the homozygous recessive, the white mice. And so what about the allele frequencies? So if we have 120 mice, twice that, 120 times 2 will give us the total number of alleles. And so you have to take account of how many brown alleles, 65 times 2 plus 50, divide that over 240, you come up with this number. For white, you take 5 times 2, that's 10, plus 50, that's 60, divided by 240, and you come up with this number here. And so now we can revisit the question we asked initially, and so we can ask, has microevolution happened in this population? And so if you go back and you look at the original allele frequency when you first went and surveyed these mice, for brown, the allele frequency was 0.7, and now it's 0.75. For white, it was 0.3, and now it's 0.25. And now you have to decide through some statistical analysis, like chi-square, if this is a significant difference, and if it's not the result of chance, if this is a significant difference, then you can say that the population is not at equilibrium, that the population is going through microevolution because the frequencies of alleles have changed over time. If the population was at equilibrium, the allele frequencies should remain real close to 0.7 for brown, if not exactly 0.7, real close to it, or for white, close to 0.3, 0.3 exactly, or slightly different from 0.3. Any deviation from that, microevolution is happening. If the allele frequencies remain the same, then the population is at Hardy-Weinberg equilibrium. And I'm just telling you, equilibrium in nature is really hard to maintain because populations are usually small, because there's going to be gene flow, individuals that are moving out in search of better places to live, because mutations have consequences on the genotype, because some individuals in the population never have a chance to mate, and when they mate, some are more successful, some are less successful, and any of those reasons will lead to microevolutionary change in a population. That's all for now, and I hope you're making good progress. Contact me if you have more questions about microevolution and Hardy-Weinberg equilibrium, and maybe I can dig out some more examples that will help you understand these concepts. That's all for now. See you next time.